All right, hello everybody. Thank you so much for coming to today's Neuroimaging Summer Series. This is our kickoff event and we're really excited to share it with you. So this series is showcasing the cutting edge research of the neuroimaging faculty at the Michigan Alzheimer's Disease Research Center. My name is Stephanie and I will be moderating things here on the back end, helping out with Q&A and the chat. So if you have questions and answers, you can see the little Q&A at the bottom and you can put your questions in there. Also, we have chat activated as well, so you are welcome to chat with us and chat with other members. Towards the end of this event, I will be sending out a Qualtrics evaluation. This is our very first neuroimaging summer series, so we're excited to get some feedback on how things are going. The evaluation will also be sent out in a follow-up email tomorrow. The follow-up email will also contain registration information on May's neuroimaging summer series, so we hope to see you all there as well. And without further ado, it is my pleasure to introduce Dr. Youjin Jung. She is currently a postdoctoral scholar in the Department of Neurology and Memory and Aging Center at the University of California, San Francisco. She received her doctoral training in the Department of Psychology and Institute of Gerontology at Wayne State University, where she worked under the mentorship of Dr. Jessica Damaso. Her doctoral research focused on the potential of plasma neurofilament light, NFL, as a biomarker of neurodegeneration for Alzheimer's disease. This research employed multimodal neuroimaging techniques and data from the Michigan Alzheimer's Disease Research Center. Her work involved close collaboration with several ADRC investigators, including Dr. Ben Hamstead, Dr. Scott Counts, Dr. Anna Doherty, and Dr. Drew Bender. Her current research seeks to identify reliable neuroimaging markers for the early detection and accurate diagnosis of frontotemporal dementia. Today, Dr. Jung will present her doctoral work, which investigated the relationship between plasma neurofilament light levels and Alzheimer's disease-related neurodegenerative changes as measured by MRI, including functional connectivity, diffusion imaging, and cortical volume. And with that, I will turn things over to Dr. Jung.
Thank you so much for the introduction. So I'm very honored to be the first speaker of this webinar series. So I will just go ahead and quickly share my screen. Can everyone see the slides? Okay, great. All right, so first of all, thank you everyone for joining the webinar today. So today I would like to share with you some of my doctoral research focusing on examining what neurodegenerative characteristics are reflected by plasma concentration of neurofilament light in older adults with and without cognitive decline likely due to Alzheimer's disease. So here's what I'd like to talk about today. So I'll first provide a brief background of my research, and I'll share 3 studies with you, which I performed to examine how plasma neurofilament light is associated with neuroimaging measures and memory function. And then I'll share the key takeaways in the conclusion. And lastly, I will take some questions. Okay, so here's some background of my research. So according to the report by Alzheimer's Association in 2023, nearly 1 in 9 Americans aged 65 and older are living with Alzheimer's disease. And the total number of people with Alzheimer's dementia is expected to increase because of the growing number of older adults. So for efficient clinical trials and improve the quality of life of patients, we need timely and precise diagnosis and prognosis, which are crucial for immediate intervention, developing tailored care plan, and medical and personal decisions. So biomarkers are crucial for diagnosis and monitoring of Alzheimer's disease. And the primary types of biomarkers for monitoring and for the diagnosis of Alzheimer's disease were markers from the cerebrospinal fluid requiring lumbar puncture or PET or MRI imaging. However, recent investigations found that some biomarkers, including neurofilament light or NFL, can be easily obtained through a simple blood draw, which is a much more accessible and less invasive way of obtaining a biomarker compared to lumbar puncture or neuroimaging. So then what's neurofilament light or NFL? NFL is one of major components of neuronal cytoskeleton, and it can be a proxy for neuronal damage in neurodegenerative diseases. And that is because when a neuron is damaged, especially particularly in the axon, NFL chains are deconstructed and released into the CSF and the bloodstream. So higher levels of NFL in the blood can be a sign of more severe damage to the neurons. And despite the advantages of blood-based NFL that I mentioned earlier, one limitation of this biomarker is that it can't tell us where the damages are located in the brain, unlike neuroimaging methods. And because NFL itself doesn't provide detailed information about neurodegenerative changes, we need to better understand what plasma NFL indicates about different aspects of Alzheimer's-related neurodegeneration, especially those revealed by neuroimaging methods, which has been the most commonly used methods to assess neurodegeneration in the human brain in vivo. And investigating this will help us accurately interpret the meanings of increased level of plasma NFL and allow us to use plasma NFL as a biomarker in optimal ways. Specifically, so we aim to investigate how plasma NFL is associated with functional connectivity and white matter microstructural characteristics in early stages of Alzheimer's disease. And we also examined whether plasma NFL is more sensitive to microstructural changes in the gray matter or white matter in early stages of ADE. So now let's discuss the first study examining the association between plasma NFL and functional connectivity. So the aim of the first study was to examine how plasma NFL is associated with the default mode network functional connectivity in older adults with and without cognitive decline. And just a quick overview of brain networks and functional connectivity. So human brain is organized into several functional networks shown in different colors here. And those networks are usually called resting state networks or intrinsic connectivity networks. The human brain is organized into these networks when you're not actively thinking about or doing something. And the regions consisting a network or a set of regions with the same color in this figure show more synchronized activities with each other. And functional connectivity refers to the degree of coherence between the activities of different brain regions. And the default mode network we are interested in today is one of such canonical resting state networks shown in red here that exhibits more pronounced disruptions in Alzheimer's disease. And I had multiple DMN or default mode network functional connectivity measures because default mode network can be further divided into multiple subnetworks, and they change in different ways in early stages of Alzheimer's disease. And so we computed these 4 connectivity measures, including posterior midline DMN or PMDMN, here shown in light blue, and connectivity between PMDMN, the hub, and temporal parietal DMN, shown in dark blue, and connectivity between PMDMN and the frontal DMN region, shown in yellow. And we also looked at the connectivity between PMDMN and bilateral hippocampus, shown in red. And the posterior midline DMN, a PMDMN, is like a hub region of the default mode network. And we additionally examined how plasma NFL is associated with hippocampal volume as well as memory function to examine if our data set replicates previously found association between NFL and these measures. And all participants in all three studies I'll discuss today were drawn from the cohort of University of Michigan Memory and Aging Project, or UMMET project, which is the primary research study at the Michigan ADRC. And our sample consisted of 46 cognitively impaired individuals diagnosed with amnestic mild cognitive impairment, and those with dementia of Alzheimer's type. And we also included cognitively unimpaired individuals, and their mean ages were around 71 years old. And most individuals in the cognitively impaired group had a CDR score of 0.5, indicating very mild dementia, or CDR score of 1, indicating mild dementia. And these two groups did not show difference in most of the demographic variables as well as NFL. However, CDR global score was higher in the CI group, cognitively impaired group, compared to the cognitively unimpaired group. Okay, so here's what we found. So figure A shows the association between plasma NFL on the x-axis and left hippocampal volume, which is on the y-axis here, by group. So here, cognitively unimpaired group is shown in blue, and cognitively impaired group is shown in red. And figure B is similar to A, but this is for just right hippocampal volume. And we found that the association between NFL and hippocampal volume significantly differed by a group in both hemispheres. And specifically, higher plasma NFL was associated with smaller right hippocampal volume in the cognitively impaired group, the red, but not in the cognitively unimpaired group, blue. And left hippocampal volume showed a very similar trend, but neither the cognitively impaired nor the cognitively unimpaired group showed statistically significant association. And we also found that the association between NFL and memory factor score also significantly differed by group. So within each group, only the cognitively impaired group, red, showed a significant association between higher plasma NFL and lower memory score, which is consistent with previous findings. And finally, here are the results on the association between NFL and the 4 default mode network functional connectivity measures shown on the y-axis. And we found that the association between NFL and functional connectivity for any of these measures significantly differed by group, as demonstrated by non-significant NFL by group interactions. Also, NFL itself did not significantly predict any of these 4 connectivity measures. However, the main effect of group on the connectivity within the posterior midline DMN, which is the DMN hub region, was significant, indicating that cognitively impaired group, red, showed lower functional connectivity connectivity within this DMN hub compared to the cognitively unimpaired group. So here's a summary of the first study. We found that higher plasma NFL was associated with smaller hippocampal volume and lower memory function, consistent with previous studies. So this result further supports previous finding that plasma NFL indicates the severity of cortical atrophy and memory decline at Alzheimer's stages with mild or very mild cognitive decline. And we found that, however, plasma NFL was not associated with functional connectivity in older adults with and without cognitive decline. So this may suggest that plasma NFL may not be sensitive enough to functional connectivity differences in early Alzheimer's disease, possibly because plasma NFL reflects neuroaxonal structural damage, because plasma NFL is mainly located in the axon, whereas fMRI Bowl signals, which are used to calculate compute functional connectivity measures, have been strongly associated with synaptic function, specifically synaptic potentials. Now let's move on to the second study examining the association between plasma NFL and white matter microstructural characteristics. Okay, so in the second study, we aim to examine how plasma NFL is associated with different aspects of microstructural characteristics across different white matter regions in older adults with and without cognitive decline. And to estimate white matter microstructural characteristics, we used an approach called neurite orientation dispersion and density imaging, called NADI. So the most commonly used method to study microstructural characteristics of brain in vivo has been diffusion tensor imaging, or DTI. And this approach models microstructure of brain tissue by measuring the diffusion of water molecules within the tissue. However, a limitation of this approach is that it only provides overall tissue properties without specific information about underlying cellular components. And NADI here, NADI is a more recently developed approach that models different cellular compartments of the brain, including intraneurite, extraneurite, and free water compartments. So what's neurites? So neurites are long and thin parts of the neurons, so such as the axons or dendrites. So intraneurite space is modeled as a space occupied by thin stick-like structures like axons and dendrites. And extraneurite space is modeled as anisotropic water diffusion like a rugby ball, depicting hindered water movements in non-neuronal cells such as glial cells. And free water compartment is modeled as unrestricted or unhindered isotropic water diffusion, so like a sphere. And using NADI, we obtained 3 NADI-based indices measures. So first, neurite density index, or NDI, indicating the volume fraction of neurites such as axons and dendrites. And orientation dispersion index, or ODI, indicating the angular variability of neurites or axons. And free water fraction indicating the volume fraction of space occupied by free water, or CSF. And these are examples of voxel-wise images of neurite density index, orientation dispersion index, and free water fraction. And for statistical analysis, we used an approach called partial least squares correlation, or PLSC. So PLSC is used to identify distinct aspects of relationships between two sets of data that consists of multiple variables. So for example, say you have two data sets, first volumes of multiple cortical regions and scores of multiple cognitive domains. And using PLSC, you can find out multiple distinct aspects of relationships between these two sets of data, which are described by separate components. And because the aim of our study was to examine how plasma NFL is associated with different NADI microstructural measures in different regions of the white matter, I used PLSC to discover distinct aspects of voxel-wise NADI indices associated with plasma NFL. And our PLSC analysis yielded 2 components that significantly explains distinct aspects of covariance or correlation between NFL and white matter NADI indices. And these 2 components explained 40 and 34% of the covariance between NFL and NADI indices, respectively. And here I want to show you the unique aspects of association between NFL and white matter NADI indices captured by the first component of our PLSC analysis. And in this color map, the color of a white matter region, or a voxel, indicates the magnitude and direction of the region's contribution to the correlation between NFL and NADI indices in the first component. So, sorry. Yeah, and the brighter the color of a region, the region is more influential to the correlation between NFL and NADI indices in the first component. And after this getting a whole brain map, we wanted to focus on regions that make more reliable contributions to the correlation between NFL and white matter NADI. And those regions included superior frontal, some anterior temporal and parahippocampal, and some parietal regions near the precuneus and inferior parietal lobule. And these white matter regions, particularly the clusters in the temporal lobe, are close to the medial temporal cortices where tau tangles begin to accumulate, which is one of major Alzheimer's pathologies. And also these parietal clusters also overlap with the regions where beta amyloid plaques begin to accumulate. And to examine which of the three NADI indices are associated with NFL in the first component, we looked at the correlation between NFL and each of these NADI indices. And we found that neurite density or NDI and free water fraction, FWF, were significantly correlated with NFL. Particularly, neurite density was highly correlated with NFL. And we further wanted to examine the direction of correlation between NFL and the neurite density and free water fraction, which were significantly correlated with NFL within the reliable regions that I presented on the previous slide. So here, cool color represents negative correlation and warm color represents positive correlation. And we found that higher NFL was associated with lower neurite density, lower NDI, indicating lower density of the axons. And this result may be attributed to the neuronal loss possibly due to tau and amyloid pathologies, considering the spatial overlap between these regions and the brain areas that are vulnerable to tau and amyloid pathologies. And we found that the direction of correlation between NFL and free water fraction was not consistent across these regions. And these opposite direction of correlation may be due to different kinds of neurodegenerational changes in the white matter that could increase or decrease free water fraction. And as mentioned earlier, NADI assumes three cellular compartments, including intraneurite compartment that consists of long cylindrical neurites and extraneurite space or compartment that includes hindered water in non-neuronal cells such as glial cells and free water compartment. And when there's neuronal loss, the volume fraction of intraneurite compartment will decrease, and relatively the volume fraction of free water fraction will increase. And this is how higher NFL can be associated with higher free water fraction. And conversely, one possible factor that might reduce free water fraction is the formation of large additional structures inside or near the axons in the white matter of older adults or patients with Alzheimer's disease. Now, often these structures are pouches filled with different materials such as organelles, amyloid precursor proteins, or fluid. And because these structures do not have cylindrical shapes, unlike the axons, it's possible that the space occupied with these structures are modeled as extraneurite space. And if this is the case, the formation of this kind of structures would increase the fraction of extraneurite space, and it will eventually decrease the free water fraction. And here's the association between NFL and white matter NADI indices captured by the second component from our PLSC analysis. And here are the regions that reliably contributed to the correlation between NFL and NADI indices in the second component. And those regions were mainly located in the centrum semiovale and in the white matter regions near the lateral ventricles, often called periventricular white matter regions. And to examine which white matter NADI indices were associated with NFL in the second component, we looked at the correlation between NFL and each NADI indices. And as you may recall, in the first component, NDI, neurite density, and free water fraction, but not orientation dispersion index, was associated with NFL. However, in this second component, neurite density index was not correlated with NFL, and but ODI was the most strongly correlated index with NFL, and free water fraction also correlated with NFL. And we further examined the direction of correlations between NFL and ODI and free water fraction which were significantly correlated with NFL in the second component. And we looked at the correlation within the reliable clusters in the second component. And here, cool color represents negative correlation, warm color, positive correlation. And we found that higher NFL was associated with lower orientation dispersion index in most of the reliable voxels. And similar to the first component, the direction of correlation between NFL and free water fraction was not consistent. Sorry. And the decreasing ODI with increasing NFL in these regions may be explained by the fact that this region is abundant in crossing fibers. So here, here's a figure from a previous study. This figure shows white matter regions with 1, 2, or 3 or more different fiber orientations. And as you can see, much of the centrum semiovale regions and periventricular white matter regions are colored in dark blue, indicating that fibers in many different orientations traverse these regions. And also, previous studies found that the vulnerability of white matter tracts to aging effects varies across different white matter tracts. So as degeneration progresses, more resistant fiber tracts are more likely to last, and more vulnerable tracts are more likely to be damaged. So this non-uniform axonal loss would eventually reduce the angular variability of white matter fiber orientations. And another explanation of decreasing orientation dispersion is that these regions, particularly periventricular white matter regions, often show higher burden of white matter hyperintensities, which has been associated with white matter damage primarily due to small vessel disease. So the negative association correlation between NFL and ODI may also reflect white matter damage due to small vessel disease. So here's the summary of the second study. So we found that the PLSE components identified 2 distinct sets of white matter regions correlating with plasma NFL, and each component revealed distinct microstructural characteristics associated with plasma NFL, which possibly originate from different mechanisms. Specifically, higher plasma NFL was associated with lower neurite density, mainly in temporal parietal white matter, which may reflect neuronal loss due to Alzheimer's-specific pathologies, such as beta amyloid and tau. And a higher plasma NFL was also associated with lower fiber orientation dispersion in white matter regions rich in crossing fibers. And this correlation may reflect non-uniform loss of white matter tracts in different orientations. And also, this association may also reflect white matter damage of vascular origin. And so in conclusion, plasma NFL appears to reflect the degree of axonal loss in white matter regions commonly affected by Alzheimer's disease at very early dementia stages. All right, so this is the last study I wanted to share with you today. So by conducting the first and second study, we confirmed that plasma NFL reflects the degree of degeneration in both gray matter and white matter. And then we questioned, then is plasma NFL equally sensitive to the degeneration of white matter and gray matter? Or does plasma NFL start capturing the change in one tissue type earlier than the other in the disease progression? And to answer that question, we conducted this third study. So our hypothesis was that plasma NFL would reflect microstructural changes in white matter earlier than in gray matter as the disease progresses. And this hypothesis was based on previous findings suggesting that Alzheimer's-related microstructural changes in white matter may begin earlier than Alzheimer's-related gray matter atrophy. And the hypothesis was also based on the fact that NFL is mainly located in the axons, so plasma NFL level would be able to capture more subtle changes in in the white matter than in the gray matter. And we try to test this idea, this hypothesis, using a path model with the moderating effect of memory as a proxy for symptom severity. Also, we chose entorhinal cortex as a gray matter area of interest because this area often exhibits pronounced damage in early Alzheimer's stages. And for the white matter regions of interest for the path model, I brought them from the second study. So I used the reliable clusters in the first component of the PLSE analysis. So they were located in the white matter near the anterior temporal and parahippocampal and some parietal and some precentral regions. And for the measure of memory performance, I used a memory composite score calculated using these 4 test scores. So we expected that memory would moderate both the relationship between plasma NFL and gray matter neurite density, as well as plasma NFL and white matter neurite density. And our more specific expectations are here. So here, red line indicates those with more memory decline, so those who have had lower memory score. And blue line represents individuals with milder memory decline, so who had higher memory score. So our expectation was that NFL for white matter, NFL level would reflect the white matter neurite density already in those with less advanced cognitive decline. So even in those with milder memory decline, the blue, higher NFL would reflect the neurite density in the white matter. However, for the gray matter, NFL would not reflect the neurite density in the gray matter until cognitive symptoms become more advanced. So this is the same model as the one that I showed you, except that age and sex were added as covariates of no interest. And also the moderating effect of memory is shown as NFL by memory interaction in this diagram. And as hypothesized, memory score significantly moderated the relationship between NFL and entorhinal neurite density. However, memory did not moderate the relationship between NFL and white matter neurite density. Instead, the main effect of NFL and memory for the white matter neurite density was significant, indicating the association between higher NFL and lower white matter neurite density, and lower memory and lower white matter neurite density. And older age was also associated with lower white matter neurite density. However, age, sex, and memory was not associated with entorhinal neurite density. And to further understand the significant moderating effect of memory on the relationship between NFL and entorhinal neurite density, I plotted a graph showing the association between NFL and neurite density in the entorhinal cortex by memory score. So here, the blue line so this is how I split the sample into those with higher memory and lower memory. So I use Youjin index that maximize the sum of sensitivity and specificity for discriminating cognitively impaired and unimpaired individuals based on the memory composite score. And this division was just for the purpose of interpretation, just for the purpose of interpreting the moderating effect, this moderating effect. As we expected, in those with lower memory function, the red, higher NFL was associated with lower entorhinal neurite density. However, in those with higher memory score, blue, NFL did not predict entorhinal neurite density. And for the white matter neurite density, I found that higher NFL was associated with lower white matter neurite density, irrespective of the memory score, which was a little different than what we expected. So here's the summary of the third study. So we found that plasma for gray matter neurite density, plasma NFL reflected entorhinal neurite density only in those with lower memory functioning, consistent with our hypothesis. And for the plasma NFL white matter neurite density association, we expected that this association would be stronger in those with lower memory functioning. However, plasma NFL reflected white matter neurite density index in those with higher memory functioning as robustly as in those with lower memory functioning. And there are a couple of facts that suggest the possibility that NFL is actually reflecting white matter changes associated with Alzheimer's pathologies in individuals with higher memory functioning. So first, a considerable portion of our cognitively unimpaired individuals were the carriers of ApoE4 alleles, which is the primary genetic risk factor for late onset Alzheimer's disease. And more than half of the cognitively unimpaired individuals reported having subjective cognitive decline. And so there's a possibility that some of the cognitively unimpaired individuals were already on the preclinical stages of Alzheimer's disease with some white matter changes. And also, our white matter regions of interest significantly overlap with brain regions that are vulnerable to Alzheimer's pathologies, specifically amyloid and tau. So therefore, the association between NFL and white matter neurite density in those with higher memory score suggests that plasma NFL might capture Alzheimer's-related white matter changes happening in Alzheimer's stages without detectable cognitive decline. So in conclusion, the differing moderating effects of memory on the gray matter and white matter suggest that plasma NFL may be more sensitive to the Alzheimer's-related microstructural changes in the white matter than the gray matter. Okay, so here is the conclusion of today's talk. So first key takeaway is that plasma NFL can indicate microstructural characteristics in white matter and micro and macrostructural changes in the gray matter in older adults with and without Alzheimer's-related cognitive decline, particularly in regions that are vulnerable to Alzheimer's disease pathology. And plasma NFL also indicates memory functioning. However, plasma NFL was not found to be sensitive to functional connectivity changes in stages of mild cognitive decline. And our finding, so we believe our finding provide knowledge that may enhance the understanding of what can be inferred from plasma NFL. And our finding underscore the potential utility of plasma NFL as a biomarker for monitoring the severity of cognitive decline and microstructural and macrostructural brain changes in the early stages of Alzheimer's disease. So this is the end of my presentation, and I want to mention that this research would not have been possible without the data generously provided by Michigan ADRC and all the support of many researchers and staff members at Michigan ADRC, especially Dr. Ben Hempstead, Dr. Scott Peltier, Ari, and Yewande. And first, also, I would like to thank my PhD advisor, Dr. Jessica Damozo at Wayne State University for all the advice and support, not only for my doctoral research project, but also for my personal growth, my growth as a researcher. And I also thank my dissertation committee members, Drs. Anna Doherty, Drew Bender, and Scott Counts, for so kindly sharing their expertise and time for my doctoral work. And I also want to thank the current and previous Connect Lab members for the support and help and advice. And I also thank the Institute of Gerontology at Wayne State for providing me with amazing training and constant support. So now I'm happy to answer your questions.
Yeah, if anybody has any questions, oh, we have one already. All right. With regard to study 1, is there a reason there is more significant atrophy on one side of the hippocampus than the other?
Yeah, good question. Yeah, let me go back to that slide. Yeah, so right. So I mentioned that we found the statistically significant association between NFL and hippocampal volume on the right side of the brain for the right hippocampal volume. However, that did not necessarily mean that there was more atrophy in the right hippocampal volume. Yeah, and the the way we see it is that these two hemispheres showed almost the same patterns of association with NFL. However, for some reasons, for example, the sample size, probably we were not able to find a significant association in the left hemisphere, left hippocampal volume. And as far as I know, there is no study showing that in Alzheimer's disease, right hippocampal volume atrophy is more severe than left hippocampal volume atrophy. Any other questions?
Right, I've got another one. How closely does plasma or serum neurofilament align with CSF neurofilament levels? Are there any considerations to bear in mind when using plasma neurofilament?
Yeah, so great question. So actually, there has been several studies that looked at whether plasma NFL and CSF NFL levels align well, and they most like all papers that I can remember found very strong linear relationship between plasma NFL and CSF NFL in many different diagnoses, including Alzheimer's disease, traumatic brain injury, multiple sclerosis. Yeah, so we can say that they correspond, they align very well. And however, there are several things we should keep in mind when using plasma NFL because NFL is a peripheral marker, so it can be influenced by many different factors, such as kidney function and brain volume, BMI, and some cardiovascular risk factors, particularly diabetes. So when we try to interpret an individual's plasma NFL level, we should keep in mind that those factors could significantly influence the person's level of plasma NFL.
Okay, do we have any other questions? All right, I have another one came through. In study 3, what was the reasoning behind selecting neurite density index as the indicator of neurodegeneration?
Okay, yeah, great question. So yeah, right, I had to skip that part in the interest of time. So, and I should go back to this slide to explain that. So this hypothesis that plasma NFL would reflect microstructural changes in white matter earlier than in the gray matter as disease progresses, based on these previous findings that Alzheimer's-related microstructural changes in white matter may begin earlier than Alzheimer's-related gray matter atrophy. And however, these studies have some limitations, have a limitation that they used different measures for white matter and gray matter. So they used microstructural measures for the white matter, such as diffusion tensor imaging measures, and macrostructural measures for the gray matter, such as brain volume, cortical volume, or cortical thickness. So I thought that the reason why they found this result may partly due to the difference in the measure they used for the gray matter and white matter. So, so I thought it would be more reasonable to use the same measures for the gray matter and white matter for my study, for our study. And the reason why we specifically chose neurite density index from NADI approach was that there is evidence that NADI, like neurite density index or NADI-based indices, are more sensitive to subtle changes compared to DTI measures or volumetric measures. Also, in some gray matter studies, they found that neurite density index is a measure that shows more consistent way of changes. So for example, orientation dispersion could increase or decrease with age and with the progression of the disease. However, in previous studies, neurite density index more consistently decreased with age or with disease progression. So that's why we specifically chose neurite density for this third study.
Great, thank you very much. Any more questions before we close out for the day? Right, if there isn't anything else, just a reminder, I put the evaluation for this event in the chat. You can click on this or you will receive it tomorrow in our follow-up email. Please join us for our next neuroimaging summer series. It'll be on May 20th at noon. And Roya Homayuni will be presenting volume measures in studies of hippocampal subfield structure, methodological considerations, and applications. So we'll see you next time. Thank you very much.
Thank you very much for joining. Have a nice day.